Hello to the third episode today. And guys, this week I'm treating you 3 episodes for my birthday week. So that's really nice of me. No, I'm kidding. I would like to start this actually on a more serious tone by giving a tribute to a great scientist called Charles David Allis, who passed away on January 8th this year, 2023. So David Allis was the pioneer in the chromatin field. He is, for example, the one who mentioned the histone writers, erasers, and readers. So very amazing publications, great work, and it's something I was always discussing with my PhD advisor. His name most of the time come up in the sense that it's really nice to see those great scientists that publish a lot of work, and most of the things they do is really good, really great science. Sometimes there's not so good science, and it's a good question to ask ourselves. So you could be a great scientist but also have published not so good papers, in the sense that the discovery is fishy, in the sense maybe you can conclude something else with the data sets they published with. But nonetheless, I think David Allis was one of the great scientists that really helped the chromatin expand on their vision and the capacity to do research on chromatin. And why I'm mentioning him, it's actually the first one of the first to identify the target of an enzyme called PRMT1, which will methylate the arginine 3 of lysine H4. The arginine 3 of histone H4. And the reason I'm mentioning it is because of the recent publication and my first publication that we did with a collaborator, which is titled Histone H4 Lysine 20 Monomethylation Enables Recombinant Nucleosome Methylation by PRMT1 in vitro. So the action of those PRMT1, so PRMT1 stands for protein arginine methylation methyltransferase. So protein arginine methyltransferase. So they will add small chemicals to the protein, which can modulate the function of the protein. And the function mode of those PRMTs were not known. And even though this is a work in vitro, it's a good hint of what could be probably happening in vivo, so in the cell. So that was a really cool story to work on. And this leads me to my last point, is working with collaborator is great. And really, I had a fun anecdote on how this happened is, so at the beginning of my PhD, on my first year, my PhD advisor told me, hey, so there's this Canadian epigenetic conference, so you can go there. So I said, why not? So I found I met really nice people there, and I found this person who works on an enzyme reaction. It reminded me of what I was doing during my undergrad, and I told them, do you use mass spectrometry? Do you use mass spectrometry to validate your targets? And they said no. I was like, you should, because this could also validate the way you're doing your experiments. And I wasn't expecting them to call me back after the conference when I went back to Montreal, and it's a group in Toronto, and they say, "Hey, can we collaborate with you? You seem to be experienced with mass spectrometry analysis of histones." And I was like, "Yeah, sure. Why not? Collaboration is fun." And indeed, it allows. Like I see it, like one of the first paper that I publish is a collaboration paper. Allows you to go faster with the publication when you have different heads working around the same project. So I would say you should also choose wisely your collaborators. So we still had some difficulty with those persons because not to say harm on them or anything, but they're not knowledgeable of the mass spectrometry. And one danger is using detergents. And as biologists, to lyse cells, we always use detergent, but this is really dangerous for the mass spec because it destroys the analytical column. And obviously, they extracted their recombinant protein and histones to create nucleosome, which is really an amazing work, by the way, to do that. They used detergent to lyse cells, and even though you wash a lot your proteins, mass spec is so sensitive, you are able to detect traces of detergent afterwards. Though we had those traces of detergent, it was really fun actually to be able to troubleshoot this aspect, so we could have the cleanest and the best data we could for them. So it was, in a way, a good challenge to take. But yeah, next time, if I have to redo a collaboration, I would say I would choose more wisely the people I do deal with, because you don't want to, waste instrument time, waste columns or whatever. But it was a very cool paper. I'm really happy about that. Everything positive is happening this week, and I'm really grateful about that. It's really a fun story, and there's two things that actually motivated me to work on that paper. The first one is that PRMTs are very cool in the way that histones have a lot of modification that could happen to them. Acetylation, methylation on lysines, arginines, phosphorylation, and other PTMs. And each of those sets of PTM have what David Allis would say, the writers and the erasers. However, for arginine methylation, somehow we don't know the demethylase on the arginine. So it's really intriguing in terms of the basic science to understand, is there really arginine demethylases for those type of marks? And so far we don't know if they exist. Probably they exist, and we still haven't discovered them yet. So this is the excitement from the basic science point of view. The other point of view is in the clinical aspect, especially for oncology. Those PRMT seems to be very promising for treatments of some of the cancers. So I'm really, as a scientist, I'm really motivated by both the basic science and the translational science. And this is why you need the nice combination of both to make great science. You cannot just say, oh yeah, we need more clinical research, translational research, but it has to go from a foundation, and foundation is called basic science. And this is a good example of a very cool story, a very fun discovery. And even David Allis would speak about those, the crosstalk between histone modification. So it's a nice way where lysine methylation speaks to another arginine methylation. And for one to happen, it needs the other. So it's really exciting, and I hope we can find the same mechanism in the cell, which will consolidate this discovery, and hopefully, by knowing now how the enzyme function and the mechanism of action, we can develop potential drugs and so on to test in clinics. But aside from the drugability future for that, it's also really rewarding to work on the basic fundamental understanding of how those mechanisms work and the histone crosstalk. So I think the title of this episode will be Following the Footsteps of a Great Scientist, David Allis, and the histone crosstalk discovery we made in this paper. So I hope you enjoyed the 3 episodes this week. I haven't released micro content on the AI episode because it was harder to edit for micro content than just me speaking to the camera. It's more interactive. It's more personal. So yeah, see you in the next week. And I don't know what we'll be discussing next week, but we'll see that. So just to summarize, it's great to be a good scientist. It's an interesting subject to say even great scientists would publish not so good papers, and would you would you be the advocate of the devil and let them lose all their credibility, or would you let that paper pass? So this was always a good conversation we have with my PhD advisor, David Allis. I hope he rests in peace. Was an amazing scientist, and he really changed the way we see chromatin. And it's really great to following from what he the experiment I did for this collaborator was really inspired from his paper back then that identified the target of PRMT1 in vivo. This time we validated in vitro, so it's really nice to see that we're coherent in our story. It's always nice when you see those publications being coherent, and the novelty is this crosstalk between two modification. On that note, I hope you have a great week, and I hope you enjoyed the 3 episodes releasing this week.